Hello everyone, good morning. Hello everyone, 4S1 and 4S6 boys. So once you're on board together with me and please type your name as attendance in the chat box. Okay. So remember, please take your breakfast before the lesson. Okay, because if you haven't taken your breakfast, you can bring along your food sitting in front of laptop or in front of your mobile phone. Okay, because today's lesson is very flexible. It's not a physical classroom, it's a virtual classroom. You can do whatever you want. But most importantly, please pay attention and do your own notes during the lesson. If you have questions, please type your question in the chat box. I will reply when I read it. Okay, so for those who are on board with me, please write your name, type your name as the attendance, and we'll start in 5 minutes while waiting for the rest to join us. Okay. Okay, so far, okay, let me check the attendance. Paul from 4F6, you are here. Good morning. Hong Liang, Jia Jun, Ziyang, Bun Shen, good morning. Anthony, Yap Chi Kit, good morning. And then Wing Shan, Shao Qian, Ziyu, Jia Xuan, Jian Jun, Jun Xian, good morning. Okay, while waiting for the rest to join our virtual lesson, let's do something, okay, that to entertain yourself. Okay, I believe we didn't go to the school for a couple of weeks. I think you missed to sing our school anthem. So I'm not asking you to stand up to sing the school anthem, but let's have a game. Okay, start from me. We type the lyrics of the school song, the school anthem, in a relay game. Okay, until the end, then we'll start the lesson. Okay, so while waiting for the others to join us, let's do something, otherwise we'll be very boring. Okay, so welcome Calvin Ng, okay, from 4S1, Jun Hao, Rezao to join us. Good morning. Have you taken your breakfast? Okay, so during this lesson, you can type anything, the chat box. Okay, so you can chat with each other if you feel boring. Okay, and you can give me some comments. It's okay because you are free to do so. Okay, let's play a game, which is the relay game, to write the lyrics of the school anthem, since we didn't sing the school song for a couple of weeks. Okay, for those who join, please type your name as attendance, because I need to take the attendance. We gather 4S1 and 4S6 two classes together. Okay, remember to take your breakfast. Okay, today's lesson is very interesting, okay, and it's quite new to you. Today's lesson, we are going to start chapter 6, which is related to cell division. I believe in Form 1, Form 2, Form 3, you never exposed to this topic, and it's very interesting. And one more thing, I want your 100% concentration, because I will mention a lot of new terms in this lesson. Okay, if you have question or you cannot listen clearly, you can say, okay, sir, can you please talk loudly so that I can hear clearly. Okay, or if you miss something, it's okay, because this video, okay, this live, even though it is live, but you can replay anytime. Okay, you can replay anytime. Let's play a relay game. To sing, not to sing, is to type the lyrics of the school anthem. Okay, start from me, because I need to spend some time to type Chinese characters. Okay, so now keep playing this game. This is a relay game. I just started the first sentence of our school anthem. Keep on typing until the end, then we'll start. Okay, so check the attendance first while waiting for you to type the lyrics. Okay, Chao Jun Xian for S6, Taiwan for S1, Mei Yongping for S6, Jun K, good morning, Shi Hong, good morning, Ong Jian Yi, Kenji Ong, good morning, Wang Jun Xiao, Yoman Hong, good morning. Okay, come on. I've started the relay game. I've typed the lyrics, the first sentence of a school anthem. 山德中学屹立邳州. Okay, the rest, come on, keep on going. Okay, Xiao Qian, thank you for your cooperation. And some more, come on, until the end, we will start the lesson because now we're still waiting for the rest to join us. We have 22 viewers only, 22 audiences. So for 4S1, we have 40 students. For S6, we have 28 students. In total, we have 68 viewers today. So now we just have 27 viewers. Okay, Hong Liang, thank you. Okay, welcome Xin Kai, good morning. Welcome Henry, good morning. Okay, so while we are waiting for the rest to join us. Okay, so now we have a relay game. Type the lyrics of the school anthem. So far until Hong Liang, 宣扬文化中西并重. The rest, come on, continue. Okay, children said it's okay. If you're unable to chat, it's okay, never mind. As long as you give your name or ask your friend to type your name as the attendance. Okay. Chin King cannot chat because he is here. Okay, it's okay. Well, good morning, Chin King. Junwei is here with us. Ryan Chin from S1. Okay. Jun S1. Okay, the rest, come on. Keep on playing the relay game because we are still waiting for the rest to join us. Okay, remind your friend. Our class should be starting at 11, but the time now is 11:07. Okay, so we start at 11:10. 11:10. Okay, come on. Okay, so while waiting for the rest, I want you to get ready with your longbow with you. Okay, the longbow and then the stationaries, which means the pencil. Okay, it's important because for today's lesson, I will teach you to draw diagrams using pencil. And okay, please use 1 or 2 minutes, okay, to get ready with color pencils. I want 4 different color pencils. 4 different color pencils. Okay, come, go to take action right now. Take action. 4 different colors. Okay, color pencils. Come on, but do not use ball pen to draw. Okay, we use pencil or we use color pencil because I want you to identify different parts with different colors. Okay, so right now, please get ready. Color pencils with different colors, at least 4 colors. Okay, right now. Come on, come on, come on. Okay, thank you, Wun Shen, for the relay. Okay, so come on, while waiting for the rest to join us, let's start play the relay game to type the lyrics of the school anthem. Okay, I think most of us, okay, many newcomers, okay, Bin Wei, good morning, Su Wenxuan, good morning, Hong Shen Kai, Chai Yi Kang, good morning, Desmond Yong, good morning. Who is MLG Master? Do you take the attendance? Do you write your name to show me your name? MS Master, who is the master? Okay, yeah, I saw it. Ng Jun Xian. Yes, Ng Jun Xian. Okay, come on. We are going to start soon. Okay, Chin Jae-won, good morning. Lin Man Han, good morning. Okay, wait, class, you are late. Every time I told you when you are late to the class, what should you say? What should you say? If you are late to my class, what should you say? Come on. The time now is 11:09. 11:09. We should start our lesson at 11 morning. We are late for 9 minutes. Come on. So please type your name while you are here. Okay, what should you say when you are late to my class? Come on, for those latecomers, please give me some response. Okay, so for this lesson, I need you to get ready with color pencils with different colors, at least 4 colors. Because in the following drawing, I want you to use different types of colors to represent different parts in the cell. Okay, come on. So for those who haven't taken your breakfast, it's okay. You can sit in front of your PC, in front of your laptop, or in front of your handphone and eat while attending the lesson with me. Okay, it's okay because it's very flexible. You can do anything you want because you're not wearing the school uniform. Even me, I'm not wearing formal, but we are on the same track in teaching and learning at the same time. Okay, so we'll start in 1 minute. Hurry up. Please get ready with the colors. Okay, it's okay. You are late, but as long as you join the lesson. Okay, so the time now is 1:11:11:11. We may start the lesson right now. So for those latecomers, okay, you can ask your friends if they cannot follow the live, they can replay it anytime, or even for you, you can replay it anytime. So we may start the lesson right now. Okay, so before starting the lesson, I want you. Okay, imagine I have a glass of water. So if your glass of water is full, I want you to pour 50%, remove 50%. Why? Because if your glass of water is full, you are unable, I cannot throw any new inputs to you. So which means you need to throw 50%. The 50% remaining is your prior knowledge. Prior knowledge is what you studied previously. And the remaining 50%, the empty portion, I want you, today I'm going to give you some input of new knowledges. Okay, so good morning, Yao Ming. Okay, Yao Ming, today got special surprise for you because you are late. So I have special surprise for you. Okay, Yao Ming. So let's start the lesson. Okay, can we start now? Okay, I'm going to share screen. Sometimes I will share screen. Sometimes I will draw on the boards. So please focus. Hopefully our lesson can end before 12:30. Before 12:30, only once in a week. Only once in a week. Okay, so I want 100% concentration. Okay, and then 50% of your prior knowledge. 50% of your prior knowledge. The other 50% is the new input. Okay, why kid, who wake it? You are late to home. You are late to the lesson. I know, Wakey, you are spending time last night to write the essay, English essay. Is it? Okay, so we'll start now. So without further ado, we will start now. Okay, I'm going to share screen. Okay, for those latecomers, please get ready with the color pencils with 4 different colors. Okay, 4 different colors. Okay, I'm going to share screen. Well, I take off my cap because my hair is very bushy now. 6 weeks cannot cut my hair, so cannot show you my hair. Unless we are in a Zoom lesson, everyone show your hair. It's unfair. I just show my hair and you're not showing your hair. Okay, so let's start with biology lesson with Tamsue. So you follow my lesson today. It's very easy to study biology. Right? Okay, so if you are ready for the lesson, come on, please type ready. If you are ready for the lesson, please type ready. Good morning, Su Yao Ming. Okay, please type ready. When we are ready for the lesson, I need at least 10 students to give me the response. Okay, if 10 students are ready, please type ready, ready, ready. Come on. Yes, Paul, we are ready. Good. Junwei, yes. Jajun, ready. Come on. Henry, come on, ready, ready. Okay, let's go. Okay, we'll start the lesson. Okay, so previously, yesterday, I asked you to prepare the truth tables in your longboard, right? Because at the same time, simultaneously, okay, I want you to do something at your place, okay, while I'm teaching you. Because not just the lesson, I want you to do some hands-on activity, okay? Because we need input at the same time, we need output. Okay, so let's start the lesson now. Today we are in chapter 6. Chapter 6 is a new chapter. Okay, life is very fascinating, right? Because, okay, let's see the first diagram. Because life starts when the sperm fertilize, okay, an ovum to form a zygote, right? So this zygote is a one cell stage. So this one cell stage will divide into a two cell stage, and then to become 4, and then 8, 16, and then form an embryo and to a fetus. And after that, the mother gives birth to the baby, okay, an infant, and then this infant will grow, okay, become a kid, become a teenager, and lastly become an adult. Oh, I told you, I mean, that is a surprise for you. So this is growth. So why? Okay, a human life started from one cell to two cells, and lastly become a multicellular organisms. Okay, as you know, we are multicellular organisms, right, human? So how many cells do we have? Can you please type the number of cells? Number of cells. Previously in chapter 2, okay, we studied about cell. I've told you, okay, the number of cells in human. Okay, come on. Anyone can give me the idea? Please type the number of cells. Okay, in the chat box, let me know you have this prior knowledge. We are multicellular organisms. Come on, 4S1, 5, 6. Okay, how many cells do we have? Okay, I will see your response. Okay, another situation, life is very fascinating. 40 trillion. No, 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 no. Okay, life is fascinating. Let's see. That is a wound. But this wound can be healed, okay, in a couple of days. But let's see. After that, okay, after a couple of days, wow, you see the knee become very smooth. Okay, so this is about life. Why? Situation A, growth, okay, growth, and situation B, recovering of wound occurs because our cell keeps on multiplying from a single cell to two cell to four cells, okay, 8, 16 until become a multicellular organism is about 30 trillion of cells. Okay, so let's see this number 2048. 2048. Okay, if you are feel free in these few weeks, you can try to download these apps in your phone. Okay, 2048, sorry, not 23, 2048, 2048 to play this game. It's quite interesting. Okay, 2048. Okay, now today's lesson is about cell division. There are two types of cell division, namely the mitosis and the meiosis. Mitosis and meiosis. So remember these two terms, mitosis and meiosis. Both of them are different. So we see the first one. Okay. Okay, so for the mitosis, the function of mitosis is to produce new cells for growth, okay, and to repair damaged tissues, okay, is to increase the number of cells, And mitosis occur in all somatic cells. Okay, somatic cells here refer to all body cells except the reproductive cell. So which means there are two types of cells which are not somatic cell. Sperm is not a somatic cell. Ovum is not a somatic cell. So all body cells except the somatic cell. Okay, so whereas for meiosis, this process is to produce gametes, which means to produce reproductive cells, the sperm and the ovum, and it occurs in the reproductive organs, in the testes, in the ovaries. So for today's lesson, we only focus on the first part, which is mitosis, but not meiosis. And next week, I will focus on meiosis. Okay, is that clear? This is the function of mitosis, and where does it occur? Okay, so let's see. There are two main stages of cell division. Okay, two main stages. Okay, the first one is a karokinesis, and the cytokinesis. These are the two main stages. So karo here refer to the nucleus, and this is during the cell division. First, it involves the division of the nucleus, followed by the division of cytoplasm, and it is referred to the cytokinesis. Okay, so previously we studied about the nucleic acid, Okay, we have DNA, RNA. So the DNA will coil with the histone protein, okay, to form a chromosome, and these chromosomes is found in our nucleus, and chromosome carries genetic information, carries genetic material. So for this chapter, we'll focus on chromosome. Okay, so remember, chromosome consists of DNA and the histone protein. So in the human cell, in a typical human cell, we have 46 chromosomes. It doesn't mean that, or in our whole body, we have 46 chromosomes. No, no, no. Which means in a human cell, within the cell, there are 46 chromosomes. So where these 46 chromosomes come from? Okay, 23 chromosomes are known as the paternal chromosomes, which means from the male parent, which means from your father. Whereas the remaining 23 chromosomes are known as the maternal chromosomes, which means these are the maternal origin from contributed by the female parents, from the father and from the mother. So collectively formed 46 chromosomes in our cell. So if a cell, if a body cell, I don't care what type of cell, if a body cell has 46 chromosomes, I mean for human, okay, if a human body cell has 46 chromosomes in it, which means this cell is known as a diploid cell, which means 46 diploid cells. So when we write the number, okay, to represent a diploid cell, we use 2n, which is equal to 46. 2n, 2 here means 2 sets. One set is contributed by the male parent, another set is contributed by the female parent. So altogether, there are 2 sets, which is equal to 46 chromosomes. So since we have 46 chromosomes, it is equal to 23 pairs. 23 pairs divided by 2. Okay, so these 23 pairs, we just take one pair to be discussed. So for each pair, we call them as homologous chromosomes. Each pair, they must be in pair. So here, there are 2 examples of homologous chromosomes, okay, left and the right. So we ignore the color, we just ignore the color. So for homologous chromosome, they are same in terms of their structural characteristics. Okay, let's see the left-hand side, they have the same size, same length. So they are homologous chromosome, the one pair. One is contributed by the male parent, another is contributed by the female parent. So they are homologous chromosome. On the right-hand side, that is another pair of homologous chromosome. One is contributed by the father, another one is contributed by the mother. Okay, is that clear? So for each chromosome, okay, now I separate these both homologous chromosomes. Okay, for each chromosome, it's made of 2 identical sister chromatids. Okay, so this sister chromatids is on the left. Okay, we focus on the purple color. The left side and the right side, they are identical to each other, and they are known as a sister chromatids. And at the center, can you see the yellow color? And this part join the sister chromatids together, to join the left part and the right part together. And this part is known as a centromere. Okay, let's see. So the left side and the right side of the chromosome is the sister chromatids. Okay, and then we see the another color one, the green color. Okay, sister chromatids. Okay, the left and the right, and the sister chromatids, they are joined at the centromere. Okay. So for chromosomes that appears in the thread-like structure, okay, chromosome is a condensed form. Okay, they are thick, they are short, and they are visible under like microscope. Okay, but in some situation, chromosome, they are in a thread-like structure, just like a thread. Okay, so this situation, we give them another name, no longer chromosome, but instead of that, we give them the name chromatin. Okay, let's see. Here we have one pair of homologous chromosomes. Okay, once the chromosomes appear in thread-like structure, so this is known as a chromatin. So chromatin, you relate pin to the body shape hint they are thread-like structure. Okay, so before entering to the next part, I want you to be clear about these three terms: homologous chromosomes. They are just like twins. Okay, twins. They are same in terms of the structural characteristics. And what are sister chromatids? So in each chromosome, there are two identical sister chromatids. Just be focused on a single person, on an individual, just like there is a pair of twins. I take one of them. So sister chromatid is the left side of the body and the right side of the body. Okay, and also chromatin. Okay, now I will go to another part. Okay, let's see. There are two types of injuries. The first one, which is the physical injuries on the left-hand side, which means the wound can be healed in a couple of days or even in few weeks. But let's say a heart is stabbed by a knife. A heart is stabbed by a knife. Can it be recovered? Okay, let's give me some comment. If you think that the heart can be recovered, okay, when it is stabbed by someone using a knife, you type yes. If cannot be recovered, okay, or cannot be healed, you type no. Okay, I want you to see. Give me some comments. Okay, we see the left-hand side. Okay, when there is a physical injuries, okay, after a few days, the wound will be healed. Okay, Paul say no. Drink warm water. Hey, you very sarcastic, Vicky. Okay. So on another situation, okay, if let's say the heart is stabbed by a knife and is bleeding, is injured, okay, a heart cannot be replaced in this situation. If bleeding is very severe, if there is a severe bleeding, okay, it cannot be replaced. Why? I will explain to you in further. Okay, why? For physical injuries, it can be healed. But for this one, the internal, especially for the heart it cannot be healed. Okay, so we need a replacement. We need replacement. Okay, so you need someone to donate the heart for you. Otherwise, your heart cannot be healed. So now I relate this to some relationship story, because in my class I will share some love stories with you. In relationships, a broken heart is more suffering than physical injuries. So biology can explain our relationship story. Okay, so this can be explained through cell cycle. So this is a new term to you, cell cycle. So on your side, please follow Tam Shen to say cell cycle. Okay, so what is a cell cycle? It's a series of events, okay, take part, okay, in the cell. So which means there is not only one thing happens, but there are a series of events, okay, take place in the cell, and whereby the cell will grow and divide into two daughter cells. So in the cell cycle, the cell is growing, and at the end, it will divide into two daughter cells. Okay, so let's see. Let's different question before I show you the next sentence. Okay, are the cell cycles of different organisms same or different? Same or different? Same or different? The cell cycles are the cell cycles of different organisms same or different? Same or different? Okay, come on. Let's write. Okay, previous. Okay, now okay, please give your comments same or different. Okay, someone commented. Okay, why kid? Why not sun cell? Good question. Because in biology, it's quite sexism. Okay, it's more on the female part. Like we have mother cell. Okay, daughter cells, sister chromatid, but there is no father cell, no brother chromatids, and no sun cell. So in the future, when I want you to change everything into new. Can you do so? Okay. Okay. So everyone sees different. Yes, we are. Everyone give the correct answer. Well done. So different organisms, they have a different cell cycle. Even in a same organism, just like human, in a same organism, different types of cell have different cell cycle. So they are not in same. I mean, now I'm focusing on the cell cycle of a same organism. I take human as an example. So the cell cycle in the human cells, different human cells are not same in terms of duration and frequency. Duration here refers to the time taken to complete a cell cycle. Frequency, how frequent a cell divides. Okay, so for a typical human cell, typical animal human cell, it takes around 24 hours to complete a cell cycle. Okay, so now in terms of frequency, some cells they divide frequently, some cells they divide in a special situation, but some cells they do not divide. Okay, boys, according to what you know, do you know any cells, human cells, that divide repeatedly? Okay, keeps on dividing, which means they divide rapidly. Can you give me some ideas or share your ideas with your friends by typing the answer in the chat box right now? Okay, thank you. Waikip, sorry. Okay, thank you. Thank you for commenting because I'm too fast. Okay, I'm 慢点. Okay, it's okay. It's okay because you can replay this video anytime. It's not an issue, no worries. You can keep on replaying before you sleep. Okay, if you miss me, you just can play. You can listen to my voice. You can see my face. Okay, come on. What types of cell, human cells, divide rapidly? Come on, give me some ideas. Share with your friends. Okay, share with your friends what type of cells keep repeating, which means, okay, sir, will you upload slide into GC? Sure, yes, I will upload this in GC, but at the same time I want you to listen because some points will not be mentioned in the slides. Okay, I will give you these points verbally. Okay, so the brain cells, okay, Henry, okay, thank you for your response. The brain cells keep on dividing. Okay, good. Bone cells. Okay, some more. Okay, the epithelial cells. Okay, so I divide this cell cycle, okay, in terms of different frequency into three categories. The first one, cells that divide rapidly, and then the second situation in when they are injured. Okay, so a cell will divide to replace the damaged cell, and some cells that do not divide at all. So for those cells divide rapidly, like skin cells, the blood cells, okay, divide if damaged. Okay, liver cells and kidney cells they do not divide. Okay, in a normal situation unless they are damaged. So the function is to replace the damaged cells. But some cells that do not divide at all, once they achieve maturity, which means once they are already differentiated to this kind of cell, they will not divide again. So for example, the nerve cells. Okay, so just now your friend, okay, I don't know who is commenting that the brain cells, okay, divide rapidly. So this brain cells refers to what type of cells? Okay, in the brain cells consists of nerve cells, the neurons. So neurons do not divide. Okay, and even muscle cells. Okay, so you will be very surprised as a teacher. Why some bodybuilders, if they go for workout or they go for gym, so they become very muscular. But the question is, muscle cells do not divide. Yes, muscle cells here refer to the skeletal muscle. We have three types: skeletal muscle, cardiac muscle. Okay, so this also explains why. If a heart is stabbed by a knife, it cannot be recovered by its own because cardiac muscle cannot divide unless this heart is replaced. Okay, or it be transplanted by a donor. Okay, replaced by a donor. Okay, so for skeletal muscle, for those bodybuilders, why? Okay, they are very muscular, even though muscle cells do not divide. Because for bodybuilders or someone is become very muscular, it is the size of the muscle increases, but not the number of the cells, Okay, so now, the muscle cell will grow, grow in terms of the size, Okay, now we go to another part, which is the cell cycle. So please get ready. This is a new thing to you. So for a cell cycle, it consists of, just now we mentioned about cell cycle, different cells, they have different duration of cell cycle or even different frequency for a cell to divide. So in a cell cycle, it can be divided into two main stages. So the first one, which is the interphase. So a cell spends around 95% of its time in this phase, interphase, whereas the rest of the time, 5% of the time, a cell is in a mitotic phase. And this mitotic phase is known as the end phase. So there are two main phases, the interphase and the mitotic phase. So now, for the interphase, it can be subdivided into three phases. So the first one, which is the G1 phase. G here means gap. We call this as gap 1 or the first gap, GAP, first gap phase, followed by the S phase. S here is referred to synthesis. Whereas G2, it is similar to G1. G here is referred to the gap 2 phase or the second gap phase. So which means in the interphase, it can be subdivided into three phases, G1, S, and G2. Whereas in the mitotic phase, the end phase, it can be divided into two subphases, which is the mitosis, and we will discuss about mitosis later. And after mitosis is followed by the cytokinesis, which is the division of the cytoplasm. So collectively, both mitosis and cytokinesis, they are known as the mitotic phase or the end phase. Okay, so now I will focus on the interphase before going to mitosis. Okay, so in the interphase, there are three phases, the G1, S, and G2. Let's see what happened in G. So G here means gap 1 or the first gap. So you also can relate G to growth, G-R-O-W-T-H, growth. So in this phase, a cell is growing with high metabolic rate. So metabolism, we studied previously in the previous chapter. Metabolism here can refers to cellular respiration, which means in this phase, while a cell is growing, its cellular respiration, the rate of cellular respiration is high. And this time, the cell synthesizes proteins and organelles, for example, mitochondria, endoplasmic reticulum for plant cell. At this stage, they will synthesize chloroplast as well. And the chromosomes still appear as chromatin. They are not in a condensed form, not in a condensed structure, which means they cannot be seen clearly under the light microscope in G1 phase. How about S phase? S means synthesis. Something is produced. So something is produced here which means it's multiplication. Everything goes multiple. So in S phase, DNA replicates. Replicates, which means there are two copies of the same DNA, two copies. And each chromosome, in this time, each chromosome has two sister chromatids joined at centromere. So we go back to G1. G1, the chromosome just appear as chromatin. There is no sister chromatid at all. But in S phase, because of DNA replicates, so each chromosome, they have two sister chromatids, and both sister chromatids, they carry the same copy of DNA, which means the left side and the right side, they have same DNA copy. and they are joined, these two sister chromatids, they join at the centromere. And at the same time, centrosome duplicates. So centrosome contains a pair, each centrosome consists of one pair of centroles. One pair of centroles. Do you still remember about centroles? The function of central is to form spindle fiber during the cell division. So which means in this lesson, I will mention about the central as well as the spindle fiber. So in S phase, I repeat, S means synthesis, which means everything is getting double, multiple. First, the DNA replicates. Okay, each chromosome has two sister chromatids. The left side and the right side, they have same DNA copy, and centrosome, which consists of centroles, duplicates. Okay, remember, in one centrosome, there is a pair of centroles, and the function of centroles is to form spindle fiber. Okay, and lastly, it is G2. So G2 and G1, they're close to each other, similar, because I told you, you relate G to growth. So in G2 phase, okay, let's see. Okay, sorry, before going to G2 phase, let's see. So in S phase, chromosomes, each chromosome has two sister chromatids, okay? Now, in G2 phase, the cell, same as in G1 phase, the cell grows at the high metabolic rate. Okay, so that we can compare with G1, the first two events are same. And at this time, the cell will accumulate energy for mitosis. So here give you the hint that mitosis, it depends on the cellular respiration because we need to generate energy for mitosis. Okay, is it clear? Now, next part, I will go to the mitosis part. Okay, but for this mitosis part, I may spend much time on it because I want to teach you on how to draw different stages. Okay, so the time now is around 11:45. Is it 11:40? So we take a short break. Okay, 4 minutes break. Okay, we take a 4 minutes break, so you can go to toilet, okay, you can drink water, do whatever you want, and set alarm. After 4 minutes, please come back. Okay, if you have question, okay, or if you miss something, you can replay this video anytime. Okay. So now 4 minutes start from right now. Okay, see you later. Okay, is everyone ready? We still have 38 minutes left. Okay, time's up. Okay, is everyone here ready for the lesson? Okay, so please get ready. Okay, so for this part, I want you to prepare your, because we need hands-on activity. Okay, we are not going to do that, conduct the experiment, impossible. Okay, so just teach you to draw the stages in cell division. So please get ready. Okay, 4 different colors of color pencils. Okay, 4 different colors. Okay, so we may start now. Okay, at the same time, I want you to get ready with your long book, okay, with 2 pages on the left and right with the tables, with the titles as well. Okay, now I'm going to share screen, okay, for the next lesson. Okay, so for this part, okay, after mentioning the first phase in the cell cycle, whereby a cell spends around 95% of its time in the interphase, whereas the remaining 5% of the time it spends for the mitosis, so also known as the Chromosomes, because we know that this term Chromosomes is a division of the nucleus. Okay, so now we enter the end phase. End phase consists of 2 stages. End phase. The first one is the mitosis, and the second one is the cytokinesis. Okay, so before going to the next part, I want you to know some terms. First, okay, you must be clear. Okay, the first one, which is the parent cell. So before a cell divides, so the single cell is known as the parent cell. Sometimes it is known as the mother cell. Okay, and there is no father cell because in biology always use the female terms. Okay, so we mother cell or the parent cell, and to produce daughter cells. Okay, another term is in the following parts we mentioned about the nuclear membrane. Okay, and the nucleus as well. So these are the parts we studied previously about the nucleus. Okay, and the first part of this lesson, I've mentioned the chromosomes, okay? And each chromosome consists of 2 identical sister chromatids, the left and the right side of the body, and the joint at the centromere. So we have chromosome, sister chromatid, and centromere. And the last one, in the following parts, we will mention about the centroles, which is the organelle that forms spindle fiber, because spindle fiber is important for cell division, okay, to divide the chromosomes. Okay, so for mitosis, it can be divided into 4 stages. So the prophase, metaphase, anaphase, and telophase. So for today's lesson, I want you to spend not for the whole day, just for 5 seconds or 10 seconds, 5 to 10 seconds. So I want you to be clear on these 4 phases. Okay, the prophase, the metaphase, and the anaphase and telophase, 4 phases. Okay, so next one, I will explain. PNAT, how to remember P. So P here, you really need to prepare. Okay, a cell is preparing for division. A cell is preparing for division. Whereas N here means middle. N here means middle, which means the chromosomes now they are aligned, or they line in a single row in the middle of the cell. N stands for middle. A here represents a part. A part means 分开, 离开, separate. They are far apart. A here means apart. And P means two, which means two daughter cells are produced. Two daughter cells are produced. PMAT. Okay, so now we focus on the first one, which is prophase. Prophase. Okay, so now get ready with the color pencils with different colors. Okay, and your logbook as well. Okay, later I'll teach you on how to draw. Now you just listen, you do. Listen, you do. Don't draw anything. And if you want to jot down the notes, go ahead. So now we have four colors. I'm using four colors. So the outer one is the plasma membrane. I'm using the black color. Okay, so I will not mention about the plasma membrane. What happens to the plasma membrane? So there are four parts with different colors. The red color refers to the nuclear membrane and the nuclear membrane and the nucleus. Whereas for the blue color refers to the chromosomes, the chromosomal behavior. What happens to the chromosomes? And the green colors are the centrols. Okay, and then for the purple color is the spindle fiber, and I will explain in terms of these different parts. Okay, shall we start now? Okay, boys. So just now I've told you in the interphase, the S phase in the interphase, the centrosomes duplicate. So each centrosome consists of a pair of centrols. Let's see. Now there are two pairs of centrols. So which means there are two centrosomes. So first, in prophase, which means a cell is preparing, is getting ready for division. So the nucleus, okay, we refer to the red color. The nucleus, in this situation, the nucleus disappears. Okay, we can't see the nucleus now. And the nuclear membrane disintegrates, which means the nuclear membrane is broken down into smaller pieces. So they use this term, integrate, break down into smaller pieces. Okay, next one. Now we see the chromatins. So the chromatins now no longer chromatins. They are chromosomes, right? Because chromatins are the thread-like structure. So the chromatins now they are condensed to form chromosomes. So they become shorter, thicker, and they are visible under light microscope. So now they become chromosomes, whereby each chromosome consists of two sister chromatids joined at the centromere. Whereas the green color one, okay, the centrols move to the opposite poles. So opposite pole here, left and right, they're opposite. Up and down, they are opposite. So remember, like south and north, east and west, opposite poles. So wherever it's okay, it's free to move, can go up and down, left or right, depends on where we view the cell, okay, from which part or which angle we view the cell. So for plants, since we know that plants, they do not have centrols, so how cellular respiration takes place in a plant cell? So I will explain later. So centrols, the function of central is to form spindle fibers. Okay, and this phase, the spindle fiber starts to form. Okay, since plant cells do not have centrols, how spindle fibers are formed? Okay, so these spindle fibers are formed at the region for plant cells. Okay, the spindle fibers are formed at the region near to the nuclear membrane. So even though they do not have centrols, but they do have spindle fiber, and they are formed near to the region near to the nuclear membrane for plant cells. Okay, so now everything clear. We have different four colors, and each color represents each part. So by this method, you'll be clear about the events take place in prophase. Okay, so now we move to the next part. Okay, the first part is prophase. So what is the next stage in mitosis? Okay, come on. I want you to recall back my lesson. So after prophase, the next phase would be, please type the answer in the chat box right now. I want to see who is the first one to give me the correct answer. Come on, come on, come on. After prophase, the next step would be, okay, I give you the hint, everything comes to the middle. Okay, it starts with letter M. Okay, at the same time, I will chat, anyone give me some comments, I don't know. Okay, come on. After prophase, the next phase would be, it starts with letter N, and what is that? So for those who are late, okay, please get ready with 4 color pencils, okay, 4 color pencils of 4 different colors, and your notebook must be with you right now. Okay, so after prophase would be the metaphase. So in the metaphase, what happened? Okay, so in metaphase, I'll just refer to the diagram. What color is missing? What color is missing in this diagram, which is the red color? So previously, the red color represents the nucleus and the nuclear membrane. Since nucleus disappears and the nuclear membrane disintegrates, okay, which means it is broken down into smaller pieces, which means we no need to mention nucleus and the nuclear membrane and more. So there's no red color. So there are only 3 colors remain. So now the centrols, okay, previously centrols start to move to the opposite pole. Now the centrols reach their destination, which is the opposite pole of the cell, can be up and down or left or right. Is it clear? Okay, now what happened to the chromosomes? Okay, now this is the okay, sorry, before the chromosome, let's see the spindle fiber. Okay, previously the spindle fiber starts to form, begin to form, and now the spindle fiber are completely formed or fully formed. And what happens to chromosomes? Okay, now everything they are lined in a single row. So we use a line, okay, to describe. So the chromosomes are aligned at the equatorial plane or the metaphase plane, which means at the middle of the cell. This part is known as the equatorial plane or the metaphase plane. Okay, boys. Before proceeding to the next phase, which is the anaphase. So for this cell, it is not a human cell, because for human cell we have 46 chromosomes. So if I ask you to draw a cell with 46 chromosomes, it really takes time. So we just use an example, which means this cell consists of 4 chromosomes only. Okay, we see the previous slide. It consists of only 4 chromosomes, which means 2n is equal to 4. It's not equal to 46 because this is not a human cell. 2n, which is equal to 4. Okay, so I have finished the metaphase. Now, the next phase will be the anaphase. A here means apart, which means separate. What will be separated? Okay, the sister chromatids will be separated in the next phase. Okay, so boys, at this metaphase, you can refer to the diagram. Okay, and refer to the diagram whereby the sister chromatids are still joined at the centromere. They are not separated at all. Okay, so now we go to anaphase. Okay, let's see. By referring to this diagram, you must have some idea before I give you the description. Okay, initially in metaphase, the chromosomes that align in a single row at the equatorial plane or the metaphase plate. Okay, both also can be accepted with the same meaning. Okay, equatorial plane or the metaphase plate. So let's see. Now, the chromosomes no longer align in a single row in the metaphase plate. You can see there is no arrangement in a single row. So now what happens to the chromosomes? So the spindle fiber, okay, we also compare the spindle fiber in anaphase with metaphase. So now the spindle fiber in anaphase, they become shorter and they contract. And because of this, the spindle fiber pulls the sister chromatids to the opposite pole. Okay, so at this time, the centromere divides. As we know, the function of centromere is to join two sister chromatids left and right. Okay, sister chromatid left and right together. But at anaphase, the centromere will divide. So once it divides, once it splits, the sister chromatids are pulled apart to the opposite pole. So pull by the shortening and the contracting of spindle fiber. So in this phase, we no need to mention about the nucleus and the nuclear membrane because both of them are not present. And at the same time, we are not going to mention about what happened to the central. Okay, so it is clear. We just mentioned about the shortening and the contracting of spindle fiber, as well as the chromosomal behavior, what happened to the chromosomes. Okay, the last phase is known as a telophase. T, I teach you to remember T means two, now becomes two parts. Okay, so by referring to this diagram, what can we see? Okay, the red color parts come back again. Okay, here, first the daughter chromosomes reach the opposite pole. Okay, so initially in anaphase, the sister chromatid. Okay, so I would like to go back to the previous slide to show you. So the sister chromatids, okay, they are pulled to the opposite pole. Okay, initially the sister chromatids are joined at the centromere, but now the centromere divides, right? And the sister chromatids pull to the opposite pole. So once they reach the opposite pole, sister chromatid no longer sister chromatid. We do not use this term sister chromatid to describe them anymore because there is no sister, no brother. They are already separated. So now we give them another new name. It's known as the daughter chromosomes. Now the daughter chromosomes reach the opposite pole. 4 at the top, 4 at the bottom, or 4 at the left, 4 at the right. So now the chromosomes, they uncoil to form chromatin, which means they are converted back to the thread-like structure known as a chromatin. Okay, in telophase, we can see the red color parts, which means the nucleolus reappears and the nuclear membrane are reformed. Okay, because in prophase, these parts disappear in prophase, and spindle fiber disappear. There are no more spindle fiber in telophase. Okay, so but we still need to draw the central in the diagram. Okay, because this is one of the organelle in the animal cell. Okay, so before moving to the next part, okay, the next part would be the cytokinesis. Now I want you to get ready with your logbook, okay, and the color pencils with you. Okay, so I will teach you on how to draw the 4 phases in mitosis. So please get ready with your logbook and color pencils. Okay, shall we go to the next part? Okay, so yesterday I asked you to get ready with 2 pages, okay, left and right, with 2 different tables. So for the blank page, which means the page without lines, is for you to draw the stages, the 4 different stages in mitosis. Okay, and then on the right hand side, the page with lines is to write the events occur in mitosis. Okay, so get ready with your logbook. Now I'm going to teach you on how to draw. Okay, first, okay, we use different colors. Up to you what color you do. Okay, what color also can. Any color will do. Okay, so for the plasma membrane, you just use pencil will do. But for all the other parts, for example, the centrals, the nuclear membrane, spindle fiber, as well as chromosomes, you use 4 different colors. So you will be clear about the events occur in the mitosis. Okay. So first of all, which is the prophase. We start from prophase. Okay. Initially, I asked you to draw a table with 8 boxes, 8 grids, right? 8 grids. So first, I want you to state the stages. Okay, start from prophase. Okay, followed by metaphase, anaphase, and telophase. So this is a time to train yourself to spell these phases correctly. Okay, the prophase, the metaphase, anaphase, and telophase. Okay, so we start from prophase. I want to erase this table first. Okay, so for the plasma membrane, for the first one, prophase, the plasma membrane. Now, you use one color to represent the nuclear membrane. So in prophase, the nuclear membrane disintegrate, which means you break them into pieces. No need to be so complete. Okay, you use one color because from this view you can't see the color. Even though I use different colors, actually I'm using the red color pen. Okay, red color pen to represent the nucleus. Okay, so the nuclear membrane disintegrate and the nucleolus disappear. Since it disappear, you don't need to draw. You can't see anything. Okay, so now what happens to the chromosome? So I use another color. I use the blue color. Okay, and at the same time you use do not use a ball pen. Don't use a ball pen. Use color pencil because we know ball pen is strictly prohibited for drawing. Okay, you can label the parts okay when you write the name. So we use color pencil. I want you to be clear. Okay, so imagine now we are using a cell 2n which is equal to 4. Which means a diploid cell. This diploid cell has 4 chromosomes, and each chromosome has 2 sister chromatids joined at the centromere. Okay, so how to draw the chromosome? So here is one sister chromatid. Okay, I made it big. I draw here. So this is one sister chromatid. Here another sister chromatid. Left and right. Join at the centromere. Just like an X. So draw 4 chromosomes. Okay, so for these 4 chromosomes, there are 2 pairs of homologous chromosomes. 2 pairs. So I draw 2 long, 2 short. The 2 long are homologous chromosomes. These are homologous chromosomes. 2 long, 2 short. Next one would be the centroles. So I'm using different colors to represent the centroles. Okay, I'm using yellow color. Okay, and at your place you can use different color. So there are 2 pairs of centroles. For an animal cell, there is only 1 pair, right? But now, during S phase, in the interphase, the centrosomes duplicate, which means there are 2 pairs. So I'm using different colors. Okay, and the spindle fiber starts to form. So you just draw the array, to represent the spindle fiber building. Okay, so the 4 parts with different colors are the first one, the nuclear membrane, and there is no nucleolus, no need to draw nucleolus. Okay, the chromosomes, we assume that this diploid cell is 2n, which is equal to 4 chromosomes, and there are 2 pairs. Each pair is known as a homologous chromosome, and there are 2 pairs of centroles. Okay, previously I taught you on how to draw the centroles, just like 2 cylinders, and they are arranged perpendicular to each other. And spindle fibers formed by these centrioles. So this is an animal cell. We are not drawing the plant cell. Okay, is that clear? So this is for for prophase in the animal cell. Okay, so now I'll move to the metaphase. Move to the metaphase. So if you cannot follow, it's okay. You can replay this video anytime. Okay, now I'm going to erase. Okay, now the centrals reach the opposite pole. You can draw up and down, or left or right. It's okay as long as this is an opposite pole. Okay, next one, the spindle fiber. Spindle fiber are fully formed. Spindle fibers are fully formed. Okay, I use my color is yellow color to represent the central, and the purple color for me is represents the spindle fiber. You can use your own color, it's okay as long as you know the parts to do. Okay, so now the chromosomes, they're aligned or they're lined in a single row in the equatorial plane or the metaphase plate. Okay, so this is one chromosomes. Okay, here another chromosomes. Okay, so this is metaphase. The chromosomes are aligned in a single row at the equatorial plane or metaphase plate. So you don't need to mention about the nucleus and the nuclear membrane. Okay, so shall we go to the next part, which is the anaphase. So in anaphase, the sister chromatids that are separated, which means they're separated apart. A, anaphase. Okay, next part would be the anaphase. Okay, if you are unable to follow my speed, it's okay. Never mind, you can replay anytime. So now, for the cell, no longer a sphere, it becomes elongated. It becomes elongated. With a little bit curve in, at the center, at the equatorial plane or the metaphase plate. Okay, so the centrals. Okay, using the color of centrals, which one you select just now? Okay, so we know that in anaphase, what happened in anaphase? The spindle fiber contract and becomes shorter. So contract and shorter. Okay. Okay, no need to draw too long because it becomes shorter. Okay, so and now the sister chromatid, okay, the centromere split and the sister chromatid are pulled to the opposite pole. So when you draw the sister chromatid, the centromere is facing to the opposite pole. Okay, so here is the centromere facing to the opposite pole in this way. Okay, on another side, same. So this is how we draw the interphase. Okay, it's clear? So use different colors. One color represents the chromosome, sister chromatid are using blue color. Okay, one color represents this spindle fiber, whereas another color represents the central. Okay, so now to the last part, which is the telophase. So for telophase, okay, I wait for you all because I believe some of you are still following. Okay, they cannot follow my speed. So while waiting for the rest to complete the drawing in interphase. So for telophase, the cell, okay, is still an elongated shape because the chromatids are really pulled to the opposite pole, so it becomes elongated. And at the metaphase plate or the equatorial plane is even curved, more curved in, okay, more curved in. So now, plasma membrane, okay, curved in, so like a shape, like a body shape, okay, with the waist, okay, the chest, the chest, waist, and the hip, or a groundnut shape. Okay. Now in telophase, what will happen? Okay. Now the sister chromatid, okay, initially we call the chromosome as sister chromatid, but now no longer sister chromatid. Okay, no longer sister chromatid. They are known as daughter chromosomes. Daughter chromosomes. So the daughter chromosomes reach the opposite pole. So this is one, two long, two short, too long, too short. And here another pole, too long, too short. Okay, remember, do not draw in this way. There is no more sister chromatid because previously the sister chromatids separated in anaphase, left and right separated. Now they become daughter chromosomes. Okay, so we track back. Initially, we use the diploid cell, which is 2n, which is equal to 4 chromosomes. Now, here there are 1, 2, 3, 4, 4 chromosomes. 1, 2, 3, 4. So now two cells with 4 chromosomes. And during this telophase, the nuclear membrane reformed. So I use red color to represent the nucleus part, and the nucleus reappears. So boys, use the left side, left page. Just now I showed you left page. For the right side of the page is for writing description. Okay, nucleus. Okay, and then the spindle fiber disappears, so no need to draw the spindle fiber. But don't forget, at each pole that is central, because this is the organelle present in the animal cell. So here comes to the end, not the lesson. Here comes to the end of the drawing of 4 phases. Okay, we start from prophase, metaphase, anaphase, and lastly the telophase. Okay, boys, if let's say, if you wish to draw the phases, okay, the 4 phases of the animals, of the plant cell. So don't forget, it has cell wall, we need to include the cell wall, and do not draw centrals, because in plant cell there is no central. And the spindle fibers are formed at the region near to the nuclear membrane. Okay, so after the drawing, I will go to the next part, which is the cytokinesis. Okay, it's the last part of this lesson, and I promise our lesson will end within 1 hour and 30 minutes. Okay, hopefully 12:30 everything will stop and you can go for your lunch later. Okay, so I give you another 30 seconds to draw the diagram. So while waiting for the rest to draw the diagram, I want you to train yourself. Okay, because in yesterday I asked you to draw two tables, one on the left and one on the right. Is it? So on the left part, there are eight grids, eight boxes. Okay, eight boxes. We start from the prophase, metaphase, anaphase, and telophase. Okay. Another side, draw once again. Draw once again. So which means I want you to practice, okay, to train yourself to draw. The first time you follow my steps. For the second time, okay, try to use pencil. Okay. From the first part to the last part, okay, everything use pencil to draw. Okay, previously, okay, this is the page of the longbow. Okay, so prophase, metaphase, anaphase, telophase. Right. So here it's empty. So draw once again. You can refer to the diagram first. After that, you draw it on your own without referring PMAT. Boys, I know everyone is very free. Maybe some of you have your homework assignment, but if you feel free during this MCO, okay, because you can try and do a lot of things like just now I asked you to install the apps 2048 to play, and you try to, okay, because you know 4S1 there are a lot of musicians, okay, so try to write a lyrics, okay, or write a song, because in Japan we have a popular song PPAP, pineapple and pen. Now you can try Mitosis song PMAP. You write the lyrics. Okay, this can enable you to remember the terms. Okay, and you share with your friends. PMAT song. Okay, so draw again. So now we'll go to another part, which is a cytokinesis. Okay, guys. So for another side of your longbow, the right hand side of your longbook, there is another table, is it? So for that table, I want you to write the events, okay, of events take place in different stages of mitosis according to different parts. Okay, in prophase, what happened to the centromere and the chromosomes, the chromosomal behavior, what happened to the nucleus and the nuclear membrane, what happened to the central, what happened to the spindle fiber. So from the beginning until the end, we keep mentioning these four parts only. Okay, so you list everything, these events into a table. So you will be clear in what stage we need to mention what events, in what stage what things are not present. Okay, is it clear? Okay, now we'll go to the last part of this lesson, which is the cytokinesis. I'm going to share screen. Please get ready. Okay, if you cannot follow, please keep on replay. You will follow my steps. Okay, so please wait a while. Okay, later, after this lesson on Thursday, please, each of you, please check your Google Classroom, because I will give you an assignment regarding to today's lesson. But it's not very difficult, it's very easy. Because according to the previous assignment, you know, all the assignment given to you are assigned for you, the easy one. So no worries, it's not an issue, and don't give me excuse, okay? So keep on, we never say no during a crisis, never stop learning during a crisis. So now I'm going to share the last part of this chapter by sharing screen. So please hold on because there is some technical problem. Okay. So the last part of this lesson is regarding to the cytokinesis. Cytokinesis. So we know that in mitosis, there are two main stages. Okay, the kinesis, which involves the division of the nucleus. Okay, by referring to the diagram, okay, you can see at the end of the mitosis, which means the telophase, there are two nucleus formed. Okay, so everyone can see the slide clearly, right? Okay, so now the next part, which is the cytokinesis. Okay, the cytokinesis in animal cells and plant cells are totally different. Totally different. So first, we start from the cytokinesis. Okay, by referring to this diagram, okay, you can refer to your textbook or any reference book because in my slide there is no detailed notes for you. I want you to pay 100% of attention, 100% of concentrations on what I mentioned, and you do your own notes. Okay, the first one. Okay, we start from the cleavage furrow. So this cleavage furrow is the part curving in, cleavage furrows. Okay, you can refer to this. Okay, this is the cleavage furrow. Okay, so in cytokinesis, the plasma membrane at the middle of the cell, which means the plasma membrane at the cell equator, constrict, constrict to form a cleavage furrow. So I repeat, for the first step in cytokinesis, the plasma membrane, okay, in the middle of the cell constrict to form a cleavage furrow. So at this point of constriction, the microfilaments. So these microfilaments are found in the cytoplasm. You can refer to the second diagram. So the microfilament at this point will constrict. Okay, constrict. And now microfilament. Can you see the microfilament constrict? And now the cell constrict. So we mentioned constriction for 3 times. The first one, the plasma membrane at the middle of the cell or at the equatorial plane constrict to form a cleavage furrow. The first one. The second one, the microfilaments constrict once again, constrict and causes the cell constrict. And now form 2 daughter cells. Okay, this is about the cytokinesis in animal cell. So you mentioned about the cleavage furrow, the microfilaments. So these microfilaments are found in the cytoplasm. Okay, so next one, which is cytokinesis in animal cell, is totally different. So there is no plant cell does not form a cleavage furrow. It's due to the presence of the cell wall. Okay, because cell wall is rigid, it will maintain the shape of the cell. So instead of forming the cleavage furrow, there are Golgi vesicles, or just simply known as vesicles will do. So the Golgi vesicles gather at the center of the cell is known as what? Which is the equatorial plane. Okay, repeat. I say once and you repeat, equatorial plane. Equatorial plane. So the vesicles gather at the equatorial plane, and this vesicle, they fuse together. Okay, initially it's the small dots, the vesicle. Now many vesicles, they join, they fuse to form a cell plate. So from step 2, the vesicles fuse to form cell plate. And now the cell plate grows upward, which means by referring to this diagram, the left grow to the left and grow to the right. So the cell plate grows outward until it fuses with the plasma membrane. Until the cell plate fuses with the plasma membrane. And now, okay, the cellulose fiber. Okay, as we know that cellulose, which is the component in the cell of the cell wall, right? So the cellulose fiber now are synthesized. Okay, the new cellulose fiber, the new one are synthesized to strengthen the cell wall. Because initially there is no cell wall at the middle, right? So now the new cellulose fibers are synthesized or are produced to strengthen the cell wall. And at the end, two daughter cells are produced. Okay, so by comparing the cytokinesis of the animal cell and the plant cell, so no matter how, at the end there are two daughter cells produced. Okay, the only difference is the front part. For animal cell, a cleavage furrow is formed when the plasma membrane pinches or the plasma membrane constricts. But in plant cell, there is no cleavage furrow. Instead of that, the Golgi vesicles, or the just simply vesicles, gather at the equatorial gland, and they fuse together to form a cell plate. And the cell plate grows outward until it fuses with the plasma membrane. And then the cellulose fiber synthesized to strengthen the cell wall. Okay, so these are the topics to be discussed in this lesson. Okay, so before moving to the end, let's do a recap, okay, of today's lesson. So in the beginning, I have introduced, okay, I need to stop sharing because I'm going to show my face before the end of the lesson. Okay, so sorry for the delay, because I will stop at 12:40 because we start 10 minutes late. We started at 10. I remember we started at 11:11, 1111. Okay, because many of you were late to the lesson. Okay, so let's do a recap. Recap means we do a summary regarding to what we studied throughout this lesson. Okay, so in the beginning, I've introduced, okay, there are two main types of division, cell division. Okay, the first one is the Chromokinesis. Okay, Chromokinesis, which is the separation or the division of the nucleus, Chromokinesis. Okay, another one is a cytokinesis. Cytokinesis involves the division of the cytoplasm. So there are two types, chiro and the cytokinesis. So next one, followed by the types of cell division. Cell division, we have two types, the mitosis and the meiosis. So for today's lesson, we fully focus on mitosis. And next lesson, next week, I will introduce meiosis. Okay, so each cell has a cell cycle. So in a typical animal cell, it spends around 24 hours to complete a cell cycle. So the cell cycle generally consists of two parts. Okay, the interphase, okay, and the end phase, or known as a mitotic phase. So for interphase, we have three subphases: the G1, the S phase, and the G2. So G here refers to gap, also relate to growth. The cell is growing, okay, at a high metabolic rate, and the cell synthesize proteins and organelles. S synthesis, which means the DNA replication, centrosome duplicates, and G2. And N phase involves mitosis and cytokinesis. Okay, so now when we go to the mitosis part, so for mitosis parts, the recap for mitosis part, there are four phases, okay, the prophase, and then the metaphase, the anaphase, and the telophase. And each phase, we need to describe the events in terms of four. The first one, what happened to the central, what happened to the spindle fiber, the chromosomal behavior, as well as the nuclear membrane and the nucleolus. And at the end is the cytokinesis. The cytokinesis in animal cell and plant cell are totally different. In the cytokinesis of animal, the cell will form a cleavage furrow due to the constriction of the plasma membrane. Whereas in a plant cell, there is a formation of vesicles. The vesicles gather at the center of the equatorial plane and they fuse to form a cell plate. So this is what we study in this lesson about mitosis and the cell cycle. So before going to the end, before end this live broadcast, if any question, please give the comment, or else you can ask in our own messenger group. So please get ready. Okay, please be ready. The next lesson is on next Tuesday. And this coming Thursday, I will upload the homework in your Google Classroom. So please check your Google Classroom on Thursday. Okay, so here comes to the end of this lesson. So goodbye, class. Thank you, class. Now everyone stand up. And what should you say at the end of the lesson? By commenting at the chat box. Okay, class, goodbye. Okay, goodbye, class. And thank you, class. See you next week.